And once the T lymphocytes have went through their positive and negative selection and have lost their receptors they no longer need, then they're going to be considered immunocompetent naive cells that are going to go to lymphatic tissues, and they're going to wait for the first time they come in contact with a specific antigen that they can react to. B lymphocytes do a similar process. I told you that. They go through also selective processes, and but they do all of this in the bone marrow. And if they do not make it through both positive and negative selections, then they're going to be basically killed off. So if they don't go through, or if they don't make it through, they're either going to undergo a process called anergy, where they basically no longer have any function, or they are going to be degraded, broken down, and they'll be gone. Once we have tolerant B lymphocytes, these guys are going to produce immunocompetent clones that then will leave the bone marrow, and they will then also move into the same tissues as the T lymphocytes. They're considered naive, and they'll wait for their first contact with some pathogen. So once the B and T lymphocytes are sitting in the lymphatic tissue, say lymph nodes, those are probably one of the most common areas. We have lymph nodes, or the spleen, or malt tissue in the respiratory tract, or the intestinal walls. And a pathogen comes along. The antigen gets into the actual either the spleen, or it goes through the lymphatic system and gets into the lymph node. It's going to cause B and T lymphocytes to undergo an antigenic process or challenge. And T lymphocytes will respond by attaching to a body cell that has been infected, whereas B lymphocytes will immediately respond to the pathogen itself. And so when we're looking at helper Ts, what happens is helper Ts attach to antigen-presenting cells that are showing MHC class II proteins with non-self. And when they attach and they realize, hey, this is my pathogen that I can attack. And so they're going to start releasing chemicals. The chemicals they release are going to activate a lot of different processes. And so here we have an antigen presenting cell. Here we have the T cell, the T helper. T helper cell binds, attaches, binds, and immediately started secreting chemicals. The chemicals they secrete, interleukin-2, is going to co-stimulate itself. It also releases interleukins-2 and some other interleukins that stimulate other processes, okay? So the T cell then is going to get activated. So its first encounter is right here, and then it gets co-activated by interleukin-2. And that causes the cell to proliferate into multiple clones of itself and differentiate into either activated helper Ts or memory helper Ts. So T lymphocytes that are cytotoxic Ts are going to bind to MHC class I molecules with non-self associated. And when they bind, that's the first activation. Then the interleukin-2 from the helper T cells is going to co-stimulate, causing the cytotoxic T lymphocyte to proliferate into multiple clones and differentiate into either the killer T's or the memory cells. So activated cytotoxic T or activated killer T's and memory T's. And so here you can see antigen, MHC class 1 proteins with self, non-self associated. A cytotoxic T cell binds. It gets co-stimulated by interleukin 2 from helper T cells, and that's going to cause it to produce clones of itself. Some are going to be the activated cytotoxic T's, and some will be memories. B-lymphocytes, similar process. B-lymphocytes will attach to a pathogen, engulf the pathogen, and then present the pathogen on the surface of their cell. Then a T-lymphocyte, helper T, can bind, and when the helper T binds, it releases interleukin 4, stimulating the B-lymphocyte to proliferate and differentiate into plasma cells and memory cells. Plasma cells are what secrete millions of antibodies every minute. And so here we have a naive B lymphocyte, comes across a pathogen, brings the pathogen in, presents it on the surface of the cell. Helper T cell binds to the MHC class 2 molecule associated with the plasma cell and secretes interleukin 4. Interleukin 4 is what co-stimulates B cells, and they produce, so they proliferate into clones of themselves and then differentiate into either plasma cells or memory B cells. Helper T cells not only help stimulate your immune response, so here's humoral and cell-mediated immunity, but they also stimulate nonspecific responses. So they release chemicals that cause fever. They release chemicals that cause an inflammatory response. They release chemicals that stimulate the complement system from to occur. So helper T's really cause the majority of effects that are produced by your immune system, which is why any type of disorder that affects your helper T's is going to wipe out your immune system. Cytotoxic T cells destroy self cells, so that's over here, that have non-self associated. And then plasma cells, which would be right here, produce billions of antibodies that flood our system and inactivate the pathogens. I'll let you look up this one on your own. You can just look this up. It's not going to play. Just in your Google bar, write cytotoxic T cell activity against T cell target cells, and then put McGraw-Hill, and it'll pop up, okay? This is another animation that I want you to watch. I have posted a paper for you to answer questions. It's a 5-point activity. It's due on October 16th, I believe. That's the proper date. Let me look. It'll be like Monday night, I believe. Sorry. I just want to make sure I'm correct on the date. There we go. Yeah, Monday night, the 16th at 11:59 p.m. So you have this done when you come into class on Tuesday, okay? And you can bring it in on Tuesday. Now we're going to talk about what antibodies actually do. So antibodies are what are produced by your B lymphocytes. And antibodies have certain characteristics that always stay the same. They are composed of 4 proteins. 2 proteins are called heavy chains, and 2 proteins are called light chains. The constant region of an antibody always looks the same. The variable region of an antibody is going to be what changes depending on what that antibody binds to. And so this is what I just said. The variable regions are the regions that will bind to an antigen and respond to that antigen or cause different effects to the antigens, okay? We have multiple types of antibodies: IgG, IgM, IgA, IgD, and IgE. I don't expect you to know every single antibody and the function of each type of antibody. What I do want you to understand is that each of these antibodies has its own constant region and its own variable region. Here's the variable region, and again, here's the variable region. This is the constant region. And this is similar. This antibody, generic antibody, looks just like an IgG antibody, immunoglobulin G antibody. So antibodies work by attacking the antigen directly, and what they do, they don't kill. They're just proteins, but they bind all around the antigen, and they eliminate the ability of the antigen to cause problems. So they cover the region of the antigen, causing neutralization, so it no longer can cause any damage. Because antibodies can bind to more than one antigen, they can cause clumping. And just like I said before, but they another thing they can do is they can take things that are typically soluble in our body fluid and make them insoluble, so they clump up. That's known as precipitation. And both the agglutination and the precipitation can lead to phagocytosis, which is a good thing. So here are three basic processes that occur by antibodies. Here's your third and final antibody-mediated immunity activity. This is an animation. I have the paper on your Canvas page in the modules. You're going to take the paper and answer the questions, and at the same time go through this video, okay? I'm going to stop here, and I'll finish this chapter in the next video, okay?